So we're going to finish up what we didn't talk about with osmolarity and tonicity and a little bit of transport. And then we're going to move in directly to talking about neurons. So this exam, this overarching unit, is really about function of the brain, function of neurons, function of synapses. So how we're sending signals and coordinating responses in our body. Yeah. But we are going to talk one last time about transport, because understanding transport is important to understanding how our neurons function and how our other systems of the body function. So it's important that we get that down pat first. So where we left off, we had gone over the difference between osmolarity and tonicity. So remember that osmolarity is a number that tells you everything that's dissolved in water. We can convert that number to a pressure because osmolarity really exerts a pull on water. So if you have higher osmolarity, that means you have more solutes dissolved in the water, which means that the water wants to move towards them in order to water that space down so that everything's evenly distributed. When we were talking about osmolarity, we remember that whenever we're talking about something that's ionically bonded, that's going to fall apart in water. Those two ions are going to dissociate. So we basically double our concentration numbers for those salts, basically. And we talked about how tonicity is a little different. It's a very related concept. But when we're talking about tonicity, we're not talking about everything dissolved in the water. We're just talking about impermeable solutes. Impermeable solutes are the things that can't cross the cell membrane. So remember that the cell membrane is semi-permeable. Some things can get across, some things can't. So everything is part of osmolarity, but only our impermeable solutes are part of tonicity. So we looked at this diagram of two things that are initially isoosmotic, but this cell is not isotonic because only these purple particles are impermeant. So if we wanted to look at just the stuff that can't move across the membrane on its own, which is going to really affect the movement of water because this purple particle can't spread out across the membrane on its own. The only way we can decrease that concentration is by adding water. So these two substances, the ISF, or the intracellular fluid and the extracellular fluid here, are not isotonic. So we saw what would happen next after this first step. Our yellow solutes, our urea, could spread out because the membrane is permeable to them, but these purple solutes can't. So as the urea spreads out, this increases the total concentration of solutes on the inside of the cell. So now our osmolarity goes up, and the water then follows this osmotic gradient, this gradient of water following this pull of those solutes. So that osmotic pressure is moving the water here into the cell. Okay. So this is just a chart from your book about quick definitions of all our different terms here. An additional thing that we want to think about when we think about the movement of water and the distribution of solutes is the influence of the size of a cell or the cell volume. So here we can see in part A, we start with a cell volume of VO. So that's our original volume. And we can see that we're starting out with a high concentration of solutes inside our cell. So we can see this is 300 milliosmoles. So this is a hypotonic solution because we have fewer solutes outside, more inside. So as we have water moving, this cell is expanding. So we can see that it actually basically doubles in size here in order to bring that concentration of solutes inside down so that we're in equilibrium with the fluid outside the cell. And we can see the opposite thing happen as well. So if we're in a hypertonic solution or a hyperosmotic solution where we have a greater concentration outside the cell, a lower concentration inside the cell, we're going to have water move out of the cell in order to really increase that concentration inside to be at equilibrium. We want these both to have the same osmolarity. And we can see that this shrinks the cell. So we can see that this cell is half the size in volume to get those osmolarities to match. So as we shrink the volume of the cell, we basically up the concentration inside that cell. So remember that the word for this happens to be crenation, right? It's a thing that happens with osmosis. Okay.
I have a really quick question.
Mm-hmm.
Is it called for both double, making it bigger and smaller?
No, for getting it bigger, we're eventually going to end up with lysis. The cell will burst. Okay. So this is just going back. I don't have the slide up in here because I deleted the stuff we went over previously, but you'll see a slide on the different shapes of cells. So you can look back at that.
Thank you.
Mm-hmm. Any other questions about the osmolarity and tonicity? Okay, so we're now going to put all the pieces of transport together to look at a cell in the lining of the gut tube. So we're going to be thinking about epithelial cells. We're going to think about their polarity. We're going to think about how this allows them to absorb stuff and to secrete stuff. So the first example we're going to go through, if you took bio of the cell last year, will hopefully be familiar to you. We're going to go through all of it new here, but I know some of you have seen this specific cell before. So what we're looking at here is a cell lining the gut tube. So here we have the lumen of the gut tube. Here we have the blood flow. So the blood flow is by our basal membrane. So we have a basement membrane connecting all our cells. The basal membrane, here we have basolateral corners, the lateral part, just like in anatomy is at the sides. And then we have the side of the cell touching the lumen, so touching the outside of the body, which in this case is the hollow part of our gut tube where food would be going. That's our apical membrane. So what we really want to track in physiology is how we get cells to absorb and secrete stuff, and how we then get them to circulate through our bloodstream through to our different tissues. So we're going to have to look at transport across two membranes and across the inside of the cell to understand how we really have that exchange from one side of the cell to the other. Okay, so we're going to have our apical membrane, our basal membrane. We need to get across the whole cell. Quick note, when we're looking at epithelium, we remember from Phys Lab that these are cells that are designed to create barriers, basically. So we're going to have tight junctions, tightly sewing our epithelial cells together. So what's usually not going to be the way that we get things across is stuff going between cells. We're really going to have to move our particles across membranes. And that's good because that allows us to control what gets across and what direction it goes across. So we've got our apical membrane, our basal membrane, our basolateral membrane is here at the corners, where we can just kind of refer to the whole part together. We got our tight junctions gluing our cells together. Okay. So when we're talking about solute transport across the epithelium, and often when we're talking about really any transport going on in a system in the cell, we're going to start by thinking about our sodium-potassium pump. So in the epithelium, we have a sodium-potassium pump here on the basolateral membrane. What it's doing is moving a lot of sodium out of the cell. It's moving a lot of potassium into the cell. So it's going to be important to realize when we're thinking about this sodium-potassium pump that this is going to change the concentration of sodium and potassium. And as we move into neurons, we're going to see that it's actually also going to change our membrane potential, because we have 3 sodium ions moving out, but we only have 2 potassium ions moving in. So this isn't actually a direct replacement of the charges. So as our sodium-potassium pump runs, the inside of a cell is going to become more and more negative. An additional note about diagrams in your book, a quick hint, you should be able to tell what's going on in these cells often when we talk about the concentrations, but your book actually always uses red arrows for active transport and green for passive transport. So that's also useful to note here as we're trying to learn these systems. Okay, so we have our active transport of sodium and potassium. It's primary active. We can see that we're using ATP to fuel this. So we need to put in energy. They're both moving against their gradients. And what we're doing as we run this sodium-potassium pump is we're creating a sodium concentration, a difference in sodium across the membrane. So we have high sodium concentration here in this interstitial fluid, low sodium concentration inside the cell. And we're doing the reverse with our potassium. So remember that the way the sodium-potassium pumps work is that they first have sodium bind to the inside, kick the sodium out, and then they have 2 potassium bind to the outside, push the potassium in. So here we're getting a lot of potassium building up in the cell, high potassium concentration, and we're depleting the potassium in the interstitial fluid. So low concentration of potassium. Okay. Other things we have happening in the epithelium are going to respond to these concentration gradients that we're setting up, basically. So on the basolateral membrane, we also have a potassium channel. So basically, that potassium is going to be able to leak back across the basolateral membrane, which means we'll have plenty of potassium to continue going through this pump. And we're going to see that we'll have sodium channels here across the apical membrane. We're also going to see some glucose carriers. So I think we can see that on our next slide. Yeah. Okay. So when we're looking at our glucose carrier, we're going to see that this glucose carrier here on the basolateral membrane is what's pulling glucose into our bloodstream. So this is really how we're getting energy out of our food, how we're getting those sugars to distribute to our tissues to break down using cellular respiration. Okay, but we want to get all the glucose from our food. We don't want to just get glucose into the cells when there's a super high concentration of glucose in the lumen. You don't want to just get cellular energy if you've just eaten a donut or something. We want to get it all the time. So what's happening at the apical membrane is not passive transport of glucose, but active transport of glucose. So we're going to pump glucose against its gradient until we get as much glucose as possible into this cell. The way we do that is that we use the sodium gradient we set up using this sodium-potassium pump. Remember, we ended up, after the sodium-potassium pump was running, with a really low concentration of sodium in the cell. So when we have this low concentration of sodium in the cell, sodium can then move passively across its gradient on the apical membrane, because we'll have higher sodium in the lumen than in the cell because we've been pumping all our sodium out. And sodium moving down its gradient is going to work to power this co-transporter, so this membrane protein, this integral membrane protein that pulls glucose with it. So our glucose is moving against its gradient, up its gradient towards this area of high glucose concentration from this area of low concentration. And the reason it's able to do that is because it's getting pulled along by sodium moving down its gradient. So this is an example of secondary active transport because we're using this sodium gradient that we set up using primary active transport. Okay. Because we are actively pumping this glucose into the cell, we're building up the glucose concentration, getting it higher and higher inside the cell. So then on the basolateral membrane, we are able to passively move glucose into the interstitial fluid and into the bloodstream because we have this high concentration inside the cell. Okay. And then the blood moves our glucose away. So basically we have the bloodstream basically deleting any glucose that is building up in the interstitial fluid. So we're never really going to reach equilibrium here because the blood just pulls the glucose along. So this is why we need to know all our different types of transport, our passive, our primary active, our secondary active, because they're all linked together like this. We talked a little bit about this before the exam as well. Okay. So these are basically going over what I just said, just writing it out for you. So when we're looking at the sodium, we have low intracellular sodium because we have this pump running. This creates a gradient moving into the cell. Sodium is able to go into the cell. This pulls through our glucose, so that creates the glucose transport. And then once we have this buildup of glucose in the cell, we can have facilitated diffusion of glucose across the basolateral membrane. Okay. We are also interested in water moving across the epithelium. So sometimes we're going to absorb water, right? When we drink, we want to absorb that water. Sometimes we'll secrete water, right? Like when we sweat. We'll also see that moving water is important in our lungs. So basically how water transport happens across epithelium is that first we're going to pump solutes into the interstitial fluid. So you can see here in red, we're pumping solutes into our interstitial fluid. And we're building up a high solute concentration here in these spaces between the cells. When we do this, when we increase our solute concentration here between our cells, we're upping the osmolarity of these spaces, which means we're also upping the osmotic pressure. Okay. So we're upping the osmotic pressure, which is going to cause a pull on water. So we're then going to have water flow through osmosis. We're going to have it osmotically pulled from the lumen into these spaces where we've built up high osmotic pressure by pumping in solutes. So we're then going to have water flowing into the interstitial fluid, basically across both membranes. So it'll flow here and then across. Usually takes the shortest path kind of here into the corners. Sometimes we have problems basically with how our different transporters and different types of transport are working. So one example of a disease that impacts transport is cystic fibrosis. So it doesn't always do this directly by affecting the proteins, although that does also happen. But what happens in cystic fibrosis is what we have happening is in a normal cell, the way we get water moving into the lungs, keeping our mucus at the desired level of thickness, is that we're pumping chloride ions and we're allowing the passive transport of sodium ions into this airway, basically. And then water follows this salt gradient, moves osmotically across to like water down this area. So this keeps the mucus in the lungs at a reasonable level. So what happens in cystic fibrosis, two things, basically. One is that our chloride ions can't get across this membrane. So we do have some effect here on the proteins in the membrane. And this means that we now don't have the same buildup of salts here in the lungs, which means that water doesn't cross as well across these membranes. As water is flowing towards areas where we have this high solute concentration. So if we're not pumping solutes across this membrane, we're also not going to move water. This makes the mucus grow thicker and thicker because there's nothing to water it down, which also has the effect of essentially making this membrane thicker. So we talked about how diffusion is affected by membrane thickness. So the mucus essentially acts as additional membrane, an additional sort of block on movement of things across this membrane in our lungs. Okay. So in this way, we don't have water movement and we'll have the thicker and thicker buildup of mucus. Our final type of transport is transcytosis. So transcytosis is different than the types of transport we've been talking about so far. Transcytosis happens for macromolecules, so big ones, basically. When we want something really big to move through our cells, we need a special way to do that. So what happens? So we have our big macromolecule particle here. You can see this kind of blue blobby thing. That's what we're looking at. What happens is that it hits the apical membrane, and then that membrane folds in. So the membrane folds in, forms a little pocket. That pocket breaks off. It's now what we call a vesicle, which is just like a little bag that's holding that macromolecule. This half of this pathway is called endocytosis. So endo means inside. So this is bringing something into the cell. Whenever you see cyto, that's talking about a cell. So into the cell, endocytosis. Then if we also want to carry that macromolecule into the interstitial fluid and potentially into the blood, we're going to then have to move that macromolecule out of the cell. So this process of moving something out of the cell is called exocytosis, and it happens in the reverse. So we have this vesicle join in with the membrane. Okay. And when membranes contact each other, they just start to flow together. So that vesicle joins with the membrane, forms a pocket, and then just spreads out, which means everything inside of that vesicle is now deposited into the interstitial fluid as that bag basically breaks up. So when we have transcytosis, so moving across a cell from its apical membrane through the cytoplasm and then out its basolateral membrane, what we're talking about is endocytosis and exocytosis working together to travel all the way across the cell. So we are going to see these things come back as we talk about neurons in the brain, but it'll be a little while till we see transcytosis again. All right. As we're getting ready for the membrane potential of neurons, questions to this point? We're really shifting topics now. Okay. And this is just a reminder to, as we're moving forward with this unit, check the syllabus, check that schedule for the Mastering A&P assignments that go along with the chapters that we're covering now.
Quick question. I saw in the syllabus that it was chapter 7 in Mastering A&P was due today, but then if I look on Brightspace, it said it wasn't due till the 14th. Should we follow the schedule or Brightspace?
You should follow the schedule. I often leave them up leave them visible for a little extra because especially with the COVID situation, I know everybody's life is crazy, but do follow the schedule. So I guess I'm calling myself out here that sometimes I'm a little lenient on those.
Got it. Thank you.
So if you didn't do it, that's a reminder for everybody to get it done tonight. Okay. So as we're talking about neurons, we're going to be thinking a lot about our membrane potentials again, and about some concentration differences, but really we're going to be focusing in on membrane potentials, differences in voltage, which is why we tried to spend a fair bit of time around them in Unit 1. Okay. So what I'd like us to do before we get into the slides themselves is I want us to imagine a cell with a sodium-potassium pump that's working. So I want everybody to grab a piece of paper, draw me a cell. It can be a nice circular cell. I want you to draw a sodium-potassium pump on it. I want you to show me that sodium-potassium pump working. So I want you to show me what's happening to the sodium, what's happening to the potassium. I want us to think about what's happening to the membrane potential as this works. So what's happening to those charges on the whole. So this will be similar to what we were just talking about with the epithelia, but it's really going to set up our picture for how neurons are working. So it's important. Okay, so I'm going to start drawing too. And as I draw, I'm going to ask you guys to call out some things. So prepare yourselves to be brave. All right, so we got our cell. So we're starting out with our nice circular fake little cartoon cell. As we saw in histology, our real cells don't look like this, but for the purposes of class, they do. Okay, so we're also going to have our pump working. So I'm going to draw on a sodium-potassium pump. And someone tell me what's the first thing that happens when our sodium-potassium pump is working? So what's happening to the sodium?
Three sodium are leaving the cell.
Yes, awesome. So we have 3 sodium leaving the cell. What is this doing to our sodium gradient? Anybody want to tell me? Where are we building up our sodium? Inside the cell or outside of the cell? Where's our sodium now?
Outside?
Yep, our sodium is outside the cell. Awesome. So what's happening next? We got some potassium. Which direction is our potassium going?
It's going in the cell.
Mm-hmm. Our potassium is going into the cell. Does someone want to tell me how many potassium are going into the cell? So we said 3 sodium are going out. How many potassium are going in?
2.
Mm-hmm. So we have 2 potassium going in. I'm going to write onto our sodium, right? We had 3 of them. And this is important for this final question about the membrane potential. So as we're moving 3 sodium out of the cell and 2 potassium into the cell, what would you say is happening to the charge inside the cell? So pretend we start out with 0 millivolts, right? We have the same charge inside the cell and outside the cell. What's going to happen as we run this pump?
It's going to become more negative, right?
Yeah, exactly. So as we run our sodium-potassium pump, what we're doing is creating a negative membrane potential. So negative. And this is because our pump isn't transferring these sodium and potassium equally, right? We have 3 going out, 2 coming in. So if we just count those charges, it's not even. When we're talking about membrane potentials for cells, we've simplified it in our previous unit as thinking about just a bunch of anions inside the cell and cations outside the cell. But what we're really doing is comparing the inside of the cell to the outside of the cell. So the fact that we have more positive here outside the cell means that when we compare them, the inside of the cell is negative. Okay. So this is actually how we set up the membrane potential of our cells. And this is why we end up with that typical measurement that we see on our figures and our examples of -70 millivolts about. Right? There's a range for different cells, different tissues, but this is our value for neurons. And this is how it gets set up, is by this sodium and potassium pump working, creating an uneven distribution of charges. So we call this sodium-potassium pump electrogenic. And we can see, and we'll see as we go through the membrane potential, and we go through each ion individually, that in addition to creating this membrane potential, we're also at the same time creating a chemical gradient, right? So we're setting up both this difference in charge and a difference in concentration. So this is going to lead to our equilibrium potentials for sodium and potassium. So before we talked about comparing equilibrium potentials to membrane potentials, but now we're going to see how they actually get set up in the first place. closing my annotation tool. Okay. So that was a really quick intro to the establishment of the resting membrane potential. But that's basically what we're going to go through in the next several slides. So we're going to be talking about how we set up this membrane potential and how that actually determines our equilibrium potential for the potassium ions and the sodium ions. Remember that our equilibrium potential is the point at which our electrical driving force and our chemical driving force are directly in opposition, cancel out to zero. Okay. And we can use that to figure out which direction our ions want to move, because when we compare it to the membrane potential, it's going to tell us something about the electrochemical driving force. Okay. So our resting membrane potential exists because there are more negative charges inside the cell and more positive charges outside the cell. So really here we want to focus on that positive charge outside the cell, right, produced by all that sodium getting pumped out and not enough potassium getting pumped in to cancel it out, right? So any negative charges that were inside the cell, when we kicked out 3 sodium, only brought in 2 potassium. Suddenly, if we weigh the balance of positive to negative inside the cell, our negative starts to win. All cells have a resting membrane potential. They're negative unless something's going on. We'll see that at certain points, our neurons are going to spike positive. We call that excited. We'll call those cells excited when they move into the positive range. But the resting membrane potential is -5 to -100. And for neurons specifically, which is the focus of this unit, our resting membrane potential is -70 millivolts. Okay. So we're going to go through potassium and sodium individually. And we're going to see that when we're creating this equilibrium potential, we're going to be tracking our concentration gradients. We're going to be tracking membrane permeability. We're going to be tracking those electrical charges. When we're looking at the resting membrane potential, which is adding up all the charges, whether it's sodium, whether it's potassium, we'll see that we need to take into account those ion concentrations, those ion forces caused by the equilibrium potential, but also whether the ions can actually get across the membrane. That's going to be really important as we look at the action potential of neurons, which is the way we're going to be sending signals as we move forward. Okay. This is from your textbook. This is a list of some of our definitions of these really important terms. So when we're talking about potential or potential difference, it's just the difference in charge between 2 points, difference in voltage. So we could hook up a voltmeter and measure that. You actually will use a voltmeter at some point in physiology lab. So you'll actually be measuring action potentials eventually. Our membrane potential is talking specifically about the voltage across the plasma membrane. So we could have a potential difference like here versus here, but our membrane potential is just on either side of the membrane. We're always giving the voltage inside the cell relative to the outside of the cell. Our resting membrane potential is just when the cell is not sending or receiving signals, it's hanging out at some membrane potential. Neurons, it's hanging out at -70. We'll go through what graded potentials, action potentials are in future lectures. So we're going to skip here to equilibrium potentials. Remember from Unit 1, our equilibrium potential is when we have a membrane potential that cancels out. So our membrane potential cancels out the chemical forces to move an ion across the membrane by having that charge that directly pulls them into the membrane with equal strength, equal magnitude. Okay. So 20% of the resting membrane potential is directly due to the sodium potassium pump. So we were drawing our cell, we were drawing that sodium and potassium ATPase just another word for the sodium potassium pump. It's just emphasizing that this is primary transport, right? Because it's telling us it's an enzyme that breaks down ATP, which is telling us that we're breaking down ATP for energy. Same thing, just another term. So 20% of our resting membrane potential is directly due to that pump. So it's electrogenic because it's moving 3 positive charges in the form of sodium out, 2 positive charges in the form of potassium in. So our net export of charges is we have a net export of 1 charge out. 80% of our resting membrane potential is actually indirectly due to the action of our sodium potassium pump, because the sodium potassium pump produces concentration gradients. So we'll see that that resting membrane potential and those concentration gradients are going to exert forces on the sodium and the potassium. So we want to remember that for the concentration, our sodium has a high concentration outside, a low concentration inside. Our potassium has a low concentration outside, because that's where we've been getting our potassium to move in. So we have a high concentration inside. So 20% is just about the charges, and then 80% is going to be using those concentration gradients and seeing those effects. So when we're looking at the typical neuron, it is permeable to both potassium and sodium. So when we say it's permeable, what I mean is that there will be channels that allow sodium and potassium to cross the membrane. Not that like we have some weird kind of membrane where potassium and sodium can get through that lipid bilayer. So there are proteins, there are these channels for facilitated diffusion, basically. But we have a difference in the number of those channels. So our neurons are going to be 25 times more permeable to potassium than they are to sodium. So potassium can get across a lot easier than sodium can. Now, we're also going to see in our neurons that we have a distribution of ions. So we already talked about the sodium and potassium part. We have a lot of sodium outside the cell, not very much potassium, and a lot of sodium, a lot of sodium outside the cell and a lot of potassium inside the cell. We're also going to have distribution of chloride and other anions. So chloride, so the chlorine ions, anions, are outside the cell, and organic anions are inside the cell. And we won't focus as much on those really. So in terms of importance, remember about the sodium and potassium. Okay. So looking at the potassium first, we're going to start with this cell that has no membrane potential. So it has no membrane potential, 0 millivolts, but it does have this high potassium concentration inside. Okay. So let's say we ran our pump, we concentrated the potassium inside the cell, we stopped running the pump, so our charge dissipated, but the potassium ions didn't dissipate. They stayed inside the cell because we're starting out here before the potassium ions move, basically. Okay. So the driving force on these potassium ions is going to be out of the cell. So our chemical force on those potassium is to move our potassium ions out. Okay, so we let them do that. So now we can see that we have some potassium ions. They've moved out of the cell while this cell is permeable to potassium. So as the potassium moves out of the cell, it becomes negative. Now we know that in fact, we had an electrogenic pump. So actually we started out with a negative, but as potassium moves down its gradient, its chemical gradient becomes even more negative. So that's that adding into that 80% of the membrane potential that we saw on a previous slide. Okay. So potassium, following its chemical gradient, moves positive charges out of the cell. And as positive charges move out of the cell, that cell is becoming more negative. So the inside of the cell becomes negative, partially due to that chemical force of the potassium. As the inside of the cell becomes negative, this does create an electrical force on the potassium ions. So those potassium ions are drawn back in, but they're not drawn back in that hard when we start out, right? Eventually the cell does reach equilibrium. So we have enough potassium ions transported out of the cell that this negative charge, as it gets more and more negative, pulls those potassium ions more and more. So eventually these two charges are equally opposing each other. So that membrane potential of the cell is just as strong as that chemical driving force pulling our potassium ions out. At that point, we've reached equilibrium and the movement of the potassium ions stops. That's our equilibrium potential. So the amount of charge we need inside the cell in order to oppose this chemical force. For potassium, it's negative 94 millivolts. So at the equilibrium potential of potassium, we have chemical and electrical driving forces that are opposite in direction, but equal in magnitude. So they're just as strong, but opposite directions. Okay. So an ion is at equilibrium when there's no net force for it to move across the membrane. Our chemical force is equivalent to the electrical force, and our electrochemical force, when we add them together, because they're in opposite directions, is equal to 0. Okay. I think we should pick up with what's going on with the sodium equilibrium potential next time. So does anybody have questions about just the potassium part? Or the sodium potassium pump? We did go over that. Okay. So I'm gonna quick summarize between what we were talking about. Oh, I can't scroll back to our drawing. So we had our sodium potassium pump work, set up a potassium gradient, which means that there's a chemical force acting on potassium. As we let channels release that chemical force, as we allow the potassium ions to move across the membrane, makes the cell more and more negative. And if we let it, it'll go all the way to its equilibrium potential where the forces would directly oppose. So if we scroll back to this slide on the sodium potassium pump, we're talking about 20% of our membrane potential having to do with the transport of charges unequally between the sodium and potassium. And then when we're talking about the potassium moving out down its chemical gradient, this is adding into the rest of that 80% of the resting membrane potential, because as potassium moves down its gradient, cell gets more and more negative. And we will see that our cell is a lot more permeable to potassium than it is to sodium. So we're going to have a lot more charges moving out, a lot more potassium moving out down the potassium gradient than we'll have sodium moving back in. All right. So as usual, I will stick around for any individual questions, but that's it for us as a big group for right now.